So, welcome to our final project for microbial biochemistry. This project is an identification of a microorganism, and we are presenting our team members Mr. Ronaldo Perez Magdaleno, Patrick Joey Martinez, and Ricardo Garcia Ruiz. On the first instance, we have the bacteria. First, the procedural results. This was the procedure used for the identification of the microorganism. From a pure culture of the given bacteria, we make a suspension in 0.85% saline solution. Then we make an inoculation in a petri dish. We make a discontinuous method strictly in a Simon citrate agar grit. Then in a test tube containing zinc sulfide in motility medium, we make an inoculation by stabbing. Then in a test tube containing the saline suspension, we add methyl red and bogus cover medium. We incubate all those test tubes in a temperature range from 35 degrees Celsius to 37 degrees Celsius for 1 or 2 days long. And here we have the results of said biochemical tests. We have the triple sugar iron test that is not applicable. The citrate test not applicable either. We have a motility test that gave a positive. The induct test is not applicable. Neither the sulfide, hydrogen sulfide, is not applicable. The methyl red test is not applicable, and the bacteria gave a positive in molecular oxygen production. Also, this was the place where the sample was taken. The the sample was taken at some thermal waters, and in the microscope, we a contrast staining was made, and the sample was seen at a times 400 magnification, and we can see that the microorganism is identified as Thermus aquaticus. The importance of Thermus aquaticus is that it's a bacteria that can withstand increased temperatures, but more than that is that the bacteria has a special enzyme called Taq DNA polymerase, and it's widely used in PCR, that is DNA amplification technique. And also, its discovery meant that various species of bacteria, especially the Thermus family, thrive in hot springs. And we can find this kind of bacteria in hot springs. The Thermus aquaticus was originally discovered in Yellowstone National Park at United States of America.
From Thermus aquaticus bacteria, different relevant biomolecules can be isolated. For example, fructose biphosphate aldolase, or commonly known simply as aldolase, which is an enzyme that catalyzes the decomposition of certain sugars and their transformation into usable energy in the process known as glycolysis. A second example is the so-called RNA polymerase 1HQM. This biomolecule can implement ribonucleotides to synthesize RNA from a sequence of DNA that serves as a pattern or model. And a last example, and perhaps the most important out of these three, is a DNA polymerase biomolecule known as Taq polymerase. It receives this name because it is obtained from Thermus aquaticus bacteria, the T meaning thermus and the AQ meaning aquaticus. What makes this molecule so important is its use in PCR, which is polymerase chain reaction tests, which can be used to identify genetic material from samples and to determine or diagnose infections or diseases. One well-known example of this test is for the detection of the infamous virus that causes COVID-19. This bacterium is a chemotroph, which means that it obtains its energy through chemosynthesis. In hot springs where Thermus aquaticus is commonly found, there is sulfur present in the water in the form of hydrogen sulfide, which can be oxidized to produce usable energy. This synthesis consists of the reaction of carbon dioxide, water, hydrogen sulfide to produce carbohydrates such as sugar and sulfuric acid. But also, Thermus aquaticus obtains food from a group of bacteria that live with it, which are called cyanobacteria, which perform photosynthesis and whose products are used by the Thermus aquaticus to obtain energy. In the hot springs where Thermus aquaticus is found, cyanobacteria can be identified by the green color in water next to a yellow color which is caused by these bacteria. The bacteria Thermus aquaticus reproduces asexually by means of binary fission. Binary fission is a way of asexual reproduction which consists of the separation of the body into two new bodies, each receiving one exact copy of DNA that was duplicated in the original body.
Now we're going to talk about the growing condition about our bacteria Thermus aquaticus. The growing of the Thermus aquaticus fundamentally depends on the pH and the temperature. The pH tag has a pH growth rate between 6 and 9.5, but the most optimal pH is between 7 and 7.5. In the temperature growth rate is between 45 and 78 degrees, but the most optimal temperature is 70 degrees. Now, in the isolation methods, we found the serial dilution for Thermus aquaticus. So in the first step, we're going to deposit 30 grams of organic materials in 270 milliliters of sterile water. Then we're going to shake it at 150 RPM for 30 minutes at room temperature. And then we're going to perform the method of serial dilutions from 10 at least 1 to 10 at least 12 in a 8 grams per liter solution of nutrient broth. Lastly, we're going to incubate the cultures without agitation at 50, 60, 65, and 70 degrees for 1 to 6 days. Now, in the deletion methods, tag can be chemically inactivated by temperature. At low temperature, tag is inactivated, and its modification can be reversed at high temperatures. The growth of tag is not good if it isn't in the range of 7 to 7.5 for pH and in the range of 65 to 70 degrees. In the growth control methods, we found the formalin sterilization. In the first step, we're going to place the material in an airtight container. The airtight container contains a case with formalin and cotton soaked in water. We're going to leave the material around 1 or 2 days, allowing the reacting to occur. And lastly, we're going to bend the sample after the elapsed time.
Now we proceed to the presentation of the fungus. First of all, the procedure results. For the growth of a fungus, we need either malt extract agar or potato dextrose agar. We can select either of those. First, we prepare either the PDA or MEA. We empty some of the agar to a petri dish. We wait for it to solidify. Then we sterilize the work environment. That is for the inoculation of the fungi. So we take an inocule of the said fungi, preferably from a pure culture. We then trigger a discontinuous streak plate methods, and then we incubate at a temperature range of 26 degrees Celsius to 30 degrees Celsius for 1 or 2 days. And we have the results of the procedure. These are photos of the macroscopic morphology in the culture, and we also have the microscopic morphology. And then we proceed to identify the microorganism. We can do this from the microscopic structure. So we identify important parts such as the conidium, guys, medulla, branch, and conidiophore. This type of structure is characteristic of the penicillium. So the identified microorganism is Penicillium spp. The importance of this microorganism is that it holds an important economic factor because it plays a central role in the production of various cheese and various meat products, but it also serves as a means of production for numerous enzymes and other macromolecules. And also, penicillium is the producer of the drug penicillin, which is an important antibiotic and one of the first. And also we have griseofulvin, which is an antifungal drug, and it has potential chemotherapeutic agent. We can find penicillium on spoiled foods, soil, decaying vegetation, compost, or wood. We can locate it as a common green moth or common blue moth.
The most important biomolecule characteristic of penicillium is the wide-known antibiotic penicillin, the first of its kind and worldwide used medicine for the elimination of bacteria. Penicillin works in a simple way. It acts directly on peptidoglycans in the cell walls of bacteria, and when a bacterium multiplies, small holes open in its cell walls. Then, new peptidoglycans fill up these holes. However, when penicillin is present, it prevents the peptidoglycans from closing the holes in the cell wall by blocking the protein structs. Then, because of these holes in the cell walls, water enters the cell from the surrounding fluids into the cell and immediately kills it. In the microscopic morphology of penicillium, septate hyaline hyphae with a diameter of 1.5 to 5 micrometers can be seen, with branched or unbranched conidiophores. The first cell of the conidiophores is called stipe, and the secondary branches are known as medullae, which end up in flask-shaped phyalides. For the macroscopic morphology of penicillium, we know that it is a fast-growing colony with green color but white complete borders and cotton-like texture. Penicillium is a saprophyte microorganism. This means that it feeds out of dead or decomposing matter from the place they are growing in. They typically grow in fruits or bread, decomposing it and feeding from it, but they can also grow in cheeses, for example. Penicillium is a fungus that is capable of reproducing by three different reproduction techniques, which consist of the vegetative reproduction, sexual reproduction, and sexual reproduction. In the case of vegetative reproduction, it occurs the splitting of vegetative mycelium into two or more parts, with each part growing individually just like the parent's mycelium. Asexual reproduction of penicillin occurs by developing the sexual spores known as conidiophores that are fixed or immovable. Sexual reproduction of penicillium relies on the formation of ascospores. Penicillium can be either homothallic, which means that it has male and female sex organs in the same thallus, or heterothallic, meaning that it has male and female sex organs present on different thallus. Penicillium's male sex organs are referred to as antheridia, while ascogonia is known as the female sex organs.
Now we're going to talk about the growing condition about penicillium. The growing of the penicillium fundamentally depends of the pH and the temperature. The pH, penicillium has a pH ground rate between 3.5 and 10, but the most optimal pH is around 4. The temperature ground rate is between 20 and 30 degrees, but depending on the species, it can grow in the range of 5 degrees to 37 degrees. The isolation methods, we found the shallow scraping. The fungi can be isolated by surface scraping and plating or the sample on plates with PIA agar medium. The first step, we're going to add 100 milligrams per liter of the chloramphenicol and 50 milligrams per liter of ampicillin. Then we're going to deposit the plates in the incubator for 7 days at 30 degrees in complete darkness. Then we're going to transfer the different colonies to new plates with PDA medium until purified. And lastly, we're going to perform the micro and macroscopic characterization. The distillation methods. Penicillium can be chemically inactivated by temperature. It's the same of Thermus aquaticus. At high temperatures, 121 degrees, penicillium is inactivated and it has to be moist heat and administered for 50 minutes. Remember that penicillium only grow at 20 or 30 degrees. In the growth control method, we found two formalin sterilization, and it's the same method of Thermus aquaticus. In the first step, we are going to place the material in an air-thick container. This contains a case with formalin, a cotton soaked in water. We're going to leave the material around 1 or 2 days, allowing the reaction to occur. And lastly, we're going to bend the sample after the elapsed time. In conclusion, we learned throughout the elaboration of this work how to identify organisms from laboratory tests. Also, we learned about the growth condition and the isolation methods of Thermus aquaticus and Penicillium, as well as the elimination and sterilization methods. The objective of this work was fulfilled since it was possible to correctly identify the bacteria and the fungus that were provided to us. This is our reference, and thanks for your attention.